So in this last video, we're going to be talking about the electron transport chain. Let's start by reviewing what's happened already. So we've taken a 6-carbon glucose and turned that into 2-pyruvate. We've then converted the 2-pyruvate into 2-acetyl-CoAs. And then finally, in the citric acid cycle, we converted the acetyl-CoA into 4 molecules of carbon dioxide. We also lost 2 molecules of carbon dioxide during the pyruvate processing. So this is where all the carbons went. And during this process, we generated both ATP as well as NADH and FADH2. The ATP was produced by substrate level phosphorylation. And ATP can be used as cellular currency. But how are NADH and FADH2 used? And so if we think about the analogy for cellular currency, we can think about NADH and FADH2 like foreign currency or investments that have to be cashed in for usable ATP. And this is going to happen in the electron transport chain. So how does the oxidation of NADH and FADH2 in the electron transport chain make ATP? Okay. And it was actually a major discovery that it's not substrate level phosphorylation. So let's real quick review what substrate level phosphorylation is. This is when we have an enzyme that has an active site that binds a substrate, a phosphorylated substrate, and a molecule of ADP. And that enzyme helps catalyze the reaction whereby the substrate transfers the phosphate group to ADP to make ATP and a product, and that's released. And it's a different mechanism in the electron transport chain to make ATP, and it's called oxidative phosphorylation. And we'll talk later about what exactly that is. But first, let's talk about where the electron transport chain is. So here we have this white part of the slide. Let's say that's the cytosol, and we're looking at the mitochondria. The mitochondria has an outer membrane that separates the cytosol from the internal compartment, and they have cristae. So the cristae are the sac-like structures in the mitochondria shown here. And the cristae can be separated into 2 parts. There's the membrane on the outside of the cristae. This is the inner membrane, and that's different from the outer membrane. And then inside the cristae, if we were to slice these open, is a fluid-filled compartment that we call the mitochondrial matrix. Between the inner membrane and the outer membrane is the intermembrane space. So some locations to keep track of today is that the electron transport chain is a series of proteins and other molecules that are embedded into the inner membrane. So the part that makes up, the membrane that makes up the cristae. And the intermembrane space is we're actually going to generate a electrochemical gradient. And we'll talk about how that happens, but I just want to orient you to the locations that we're talking about. So embedded within the inner membrane is the electron transport chain. And the electron transport chain is primarily composed of proteins. And those proteins are complex 1. We call them complex 1. It's a series of proteins in this complex. Complex 2, complex 3, complex 4, and a small soluble protein called cytochrome C. And you can see from this diagram that complex 1, 3, and 4 pass all the way through the membrane, but complex 2 does not. There's also a very important small molecule called ubiquinone, or as I'll call it, Q. And this protein is relatively nonpolar, and so it can easily diffuse through the intermembrane. These proteins in the electron transport chain are actually organized by their redox potential. The redox potential refers to how strongly something can hold electrons. So what happens is NADH can transfer one of its electrons to complex 1. And NADH then becomes NAD plus, and complex 1 has been reduced because it's gained an electron. And then this complex 1 will continue to pass electrons to molecules with higher redox potential. So the way it's organized is that we have the lowest redox potential in complex 1, moving to relatively higher redox potential of complex 4, meaning this holds the electrons more tightly, and then eventually to oxygen, which has a very high redox potential. So if you transfer electrons slowly to molecules with higher and higher redox potential, this is going to release energy because the bonds are held between those electrons are held more and more tightly. So you can see that free energy becomes negative as electrons move through the electron transport chain. And so each successive bond that is created has less potential energy, and that means a small amount of energy is released in each set of reactions of electron transfers. So this is like going from a nonpolar to a polar bond, which is exergonic. And we're going to use this energy that's created by transferring electrons through this electron transport chain to actually generate a proton gradient in the intermembrane space. So here NADH is donating its 2 electrons to complex 1, and 4 protons are going to be pumped through. And then that's going to deliver it to Q, and Q is going to take it to those electrons to complex 3, and then when it's going to donate those electrons to complex 3, so you, again, when this occurs, when those electrons get shuttled to cytochrome C, 4 more protons are pumped through, and then cytochrome C transfers to complex 4, which then transfers it to half an oxygen to make water, and then 2 more protons are pumped through. Okay, so every time you have an oxidation reaction, right, it intermittently raises the potential energy of these complexes until those complexes get oxidized again, and it uses that energy released from oxidation to pump protons. Okay, and so we're now talking about oxidation, right? And we're going to use this proton gradient that's established, this electrochemical gradient, to make ATP, and that essentially is oxidative phosphorylation. Okay, so back to this picture here where we have our electron transport chain embedded in the inner membrane, and we've generated a proton gradient in the inner membrane space. We're going to use that proton gradient to make ATP through the ATP synthase. Okay, so how does this occur? Okay, we have our proton gradient, and then this proton gradient comes through the ATP synthase. There's 2 parts of the ATP synthase. There's the stalk, and then there's the knob. And when protons flow through the stalk, it creates a rotational force in the stalk, causing a conformational change in the knob, and that conformational change then converts ADP and an inorganic phosphate into ATP. So this entire process of taking energy from the electron transport chain to generate a proton gradient, so we have we've taken electron carriers, and we've used those electron carriers that have high potential energy to generate a electrochemical gradient, and then we've taken that electrochemical gradient to generate a motion within the ATP synthase, and then we've converted that back into chemical energy in the form of ATP. Okay, so this entire process is called oxidative phosphorylation. This idea that protons will flow down their concentration gradient because there's a concentration gradient established, and then that will generate the ATP, that specific part of the process is called chemiosmotic coupling. And because it actually generates a physical force, it's often referred to as proton motive force. So if you're getting a little bit confused in the definitions of what oxidative phosphorylation is relative to chemiosmotic coupling or proton motive force. Here are those definitions for you. So as I mentioned, here's a better diagram of the ATP synthase. So we have this stock complex here that's embedded in the plasma membrane, right? And protons are going to flow through the stock. It's also called F0. And this stock is going to rotate, and then this F1 complex is going to change conformation to make ATP. So the F1 complex, or the knob, is responsible for making the ATP. So in summary, what's occurred is we've used electrons moving down the electron transport chain to generate a proton gradient. So step 1, we've generated a proton gradient. We then take that proton gradient, flows through the electron or the ATP synthase down its concentration gradient, right, to create mechanical energy. So that rotation of the F0 subunit as protons flow through, or also called the stock. And then that spinning changes the conformation of the knob, or the F1 subunit. And that conformational change then phosphorylates ADP to make ATP. So we've gone from an electrochemical gradient to mechanical energy to ATP synthesis. And I look forward to seeing everyone in class. Thanks, bye.